Hello, I am Anthony Cicchini.
I am Bella Red.
I'm Kate Harris.
I'm Karina Delara.
And I'm Michael Bartlett.
No one likes to be sick or to see their loved ones sick. Now imagine a world without sickness in the future. This is possible through CRISPR.
Overall, as a team, we were able to break down into 15 tasks, which helped us stay on track and equally contribute to the group. The projects were divided up for research, but for the rest of the assignments, we all worked together to get them done. Anthony Cicchini is our climate monitor. He is covering the history and explanation of CRISPR. Bella Red is our project coordinator. She is covering the future potential. Kate Harris is going to cover economics. I'm covering the health, and Michael Bartlett is covering environmental and the annotated bibliography.
As a team, the criteria we define for the greatest engineering achievement of the 21st century contains a major contribution to people's health and the economy. We also want to consider that if the achievement is too new to currently have a large effect, it should have a significant future potential. Considering this criteria, our team chose CRISPR as the greatest engineering achievement of the 21st century.
The CRISPR system is the immune system of bacteria. It is a series of known virus DNA that is used to detect an intruder upon entering the cell of the bacteria. The Cas part of the system tracks the virus down and breaks apart its DNA, eliminating the threat. This system has been decoded and is now able to be utilized for our own genetic needs. CRISPR and Cas9 work together by targeting specific genetic code, slicing it, editing it, and stitching it back together. CRISPR isn't an old technology. Bacteria have been using it forever to protect themselves from viruses. The use of CRISPR for our own advantage, however, is new. The discovery of the Cas9 system was made in 1987, and the biggest discovery was made by Jennifer Dowden and her research team in 2012. The discovery made was how to reprogram the system and target specific sites for our choosing. In 2013, this discovery was used by Feng Zhang to perform the first genome edit.
CRISPR is actually showing promising results for the health of people by curing diseases. 3 trials that have been done on people were meant for treating cancer, sickle cell, and HIV. There's a clinical trial in Pennsylvania that's done with the first phase. The first phase is meant to show the safety of the procedure. The 3 patients don't show any side effects, and the cells that were altered are lasting up to 9 months, as opposed to days. In Tennessee, Victoria Gray was treated for sickle cell with CRISPR. Ever since her trial ended in 2019, she states that she hasn't needed to go back to the hospital. Editing of embryos has been done, but not for designing the perfect babies. Instead, it's been used for treating HIV inherited at birth. There are also other trials being done for treating HIV in adults, but so far still only done on animals. As of right now, clinical trials are still recent and are being monitored and being altered for safety and effectiveness before being released to the public as official treatments.
Another way CRISPR has been used to improve human health is through controlling mosquito populations. The genes that control flight in mosquitoes are sex-dependent, so scientists were able to edit the genes so that only female mosquitoes were inhibited from flying. Since only females bite mammals for blood, this prevents them from spreading diseases. The males, on the other hand, were largely unaffected, meaning they could continue to fly and pass on these genes to future generations of mosquitoes, preventing even more females from flying in the future. The same technology could theoretically be used to control the populations of other harmful or invasive species. CRISPR also has economic benefits. For example, CRISPR has been used to improve crops such as tomatoes, potatoes, soybeans, rice, maize, barley, and more. In particular, crops have been improved in 4 main categories: improved yield, such as increasing the size and number of crops; improved quality, such as improving the nutrients provided by the crops; and improved resistance to both biotic and abiotic stresses, such as drought and mildew resistance. These improvements can make crops more economical and reduce agricultural land use.
There are several approaches to measure the impact of new technologies on the economy. An easy number to consider is jobs created, which projections estimate 18,000 jobs in the UK specifically from genetic engineering over the next 10 years. Looking at the cost effectiveness of using CRISPR technology can also give insight into its economic impact. Currently, gene therapy development can cost up to $5 billion per drug, with initial pricing per patient costing $1 to $2 million depending on the severity. Although quite expensive, this treatment comes with a decrease in long-term costs such as pharmaceutical treatments, hospitalizations, and additional sicknesses. For example, treating severe hemophilia takes less than 2 years to reach financial payback, and treating spinal muscular atrophy takes less than 10 years. Considering that the cost of genetic engineering will go down as the technology advances, CRISPR provides a cost-effective solution to many diseases and other applications.
As you can see, CRISPR has come a long way from when it first started. A future part of CRISPR would to be genetically modifying pig embryos to be organ donors for the human body. They would inject the CRISPR into the embryo, and then once the piglet is born, they would able to harvest the organs that were hormonally synchronized to the person who is receiving the organs. Another example of CRISPR in the future would be for diseases such as prion disease. This neurological disease is very harmful to mammalians and humans, but for the research, they have only worked on the mammalian side, and now they are trying to work on the human side. Along with this is the stem cell therapy, and for both of these, they are looking for a more therapeutic way of helping the patients and a longer lasting result.
This technology does not come without drawbacks, though. CRISPR opens the door for genetically modified humans. Although this could improve humans in many aspects, it also comes with many moral issues over what modifications are fair and ethical. Additionally, there are fears that this technology could be restricted based off of economic class, leading to a future where there's a genetically superior upper class and a genetically inferior lower class.
CRISPR is the greatest engineering achievement because it creates cost-effective crops that yield more for the rapidly growing population, more cost-effective genetic editing when compared to other methods. It can improve the lives of millions of people by ending genetic diseases, as well as the transmission of these diseases. It creates high-skilled jobs and shows monumental future potential as the technology develops.